Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my channel SLOs of Biology. Yes, dear students, अब से biology आप पढ़ेंगे SLOs के साथ. As you know, SLOs are students' learning outcomes, and why these SLOs are important because they clearly describe what students should know and what students should understand, and what they are able to reproduce at the end of the lecture. Today's topic is Krebs cycle. So, students, आइए देखते हैं कि what is the relevant SLO with this Krebs cycle. As you know, Krebs cycle that is the part of Arabic respiration. In Krebs cycle, it was discovered by Sir Hans Krebs. And in this Krebs cycle, complete oxidation of acetyl coenzyme A takes place. So let's see the SLO of the Krebs cycle, which is outline the steps of Krebs cycle. So dear students, outline means you have to give main steps in order with brief explanation. आपने इस Krebs cycle को brief explanation के साथ order में explain करना है. I have divided this Krebs cycle into 3 steps. आप इस Krebs cycle को आसान तरीके से समझने के लिए 3 हिस्सों में divide कर सकते हैं. First, you must know the simplest outline of the Krebs cycle. Second is what are the headings of these Krebs cycle whole process. And the last step is complete Krebs cycle with energy molecules. आइए शुरू करते हैं Krebs cycle. This is the simplest outline, which is the first step. If you are looking this simplest outline, so you must understand how to draw this simplest outline. जी हाँ स्टूडेंट्स, सिंपलेस्ट आउटलाइन को ड्रॉ करने के लिए आपको इन सारे कंपाउंड्स के नेम याद होने चाहिए बिकॉज मेमोराइजेशन इज द पार्ट ऑफ लर्निंग प्रोसेस. So if you are looking, the first compound is citrate, second is isocitrate, alpha ketoglutarate, succinyl coenzyme A, succinate, fumarate, malate, and the last is oxaloacetate. इस पूरी आउटलाइन को समझने के लिए अगर आप ये नेमोनिक या ये सिंपल सेंटेंस याद करने में कामयाब हो जाते हैं, यू कैन ईजीली ड्रॉ द आउटलाइन ऑफ Krebs cycle. And the sentence is, can I keep selling snacks for money, officer? So you can see I have highlighted the CIKSSFMO, which are explaining what the compound's name of this simplest Krebs cycle. So C stands for citrate, I stands for isocitrate, alpha ketoglutarate, succinyl coenzyme A, and succinate, fumarate, malate, and in the last, oxaloacetate. Right, students? Now come to the second step of this Krebs cycle, which is synthesis, dehydration, hydration, oxidative decarboxylation, and addition of coenzyme A, synthesis of substrate level ATP, dehydrogenation, hydration, and dehydrogenation. Students, आपने ये steps बहुत ध्यान से याद करने हैं. So to understand these terminologies, you must know the meaning. Like synthesis means combination. Dehydration means removal of water. Hydration is addition of water. Oxidative decarboxylation is what if I explain, decarboxylation is removal of carboxylic group in terms of carbon dioxide. And oxidation is removal of hydrogen. And then you are looking what the dehydrogenation. This is also a terminology. It means removal of hydrogen. So this is the second step which you can prepare easily to understand the Krebs cycle. Now move towards the third step, which is the explanation of Krebs cycle. So here we have to explain also the energy molecule formation. As you are looking on my screen, the first step is synthesis. So what type of compound is synthesized? Citrate. Actually, what happens, how citrate is formed? The 2-carbon compound is acetyl coenzyme A. It will combine with 4-carbon compound, which is oxaloacetate compound. So this 4 plus 2, these 2-carbon compounds, when they combine, so they will synthesize this compound, which is 6-carbon compound. This is citrate. Then move towards the next step, which is what? The dehydration. Dehydration is what? The removal of water molecule. So in this step, cis aconitate is produced, which is what? The intermediate compound. The actual compound is isocitrate. First compound is citrate, second compound is isocitrate. Cis aconitate is intermediate compound. Second step is dehydration, third is hydration. Here removal of water molecules takes place, here addition of water molecules takes place. So after the addition of water molecules, what happens? Actually, isocitrate and citrate, these are isomers. So here you can see the 6-carbon compound is produced. After the formation of isocitrate, oxidative decarboxylation process takes place. As I have told you in the beginning of this step, जब भी आप Krebs cycle को डिटेल में एक्सप्लेन करेंगे, oxidation is removal of hydrogen and decarboxylation is removal of what? The carbon dioxide. So here from the 6-carbon compound, it is converted into 5-carbon compounds along with the loss of carbon dioxide, or we can say along with the loss of 1-carbon compound. And here what you are looking on the screen, 1 NADH energy molecule is produced. Now move towards the 5th step, which is oxidative decarboxylation along with the addition of coenzyme A. So here students, what happens? The 5-carbon compound is converted into 4-carbon compound, which is alpha ketoglutarate is converted into succinyl coenzyme A. So as I have told you, coenzyme A is added here and oxidation as well as decarboxylation process takes place here also. Again here NADH molecule is produced and 1 carbon dioxide molecule will be lost, means 1 carbon is lost. That is why 5 carbon is converted into 4 carbon. So now move towards the next step, which is the synthesis of substrate level ATP. So at this step, succinate is produced. So obviously coenzyme A is removed and obviously enzymes, they are properly, what I have to say, working in every step, right students? So enzyme is succinyl thiokinase, which is working to form succinate, and succinate is what? This is 4-carbon compound. And students, after the formation of succinate, which is a 4-carbon compound, the ATP or GTP is also released, right? It means succinyl coenzyme A is produced by the addition of coenzyme A, and then ATP is produced, which is energy molecules. Sometimes GTP is also produced. So must keep in your mind when we are discussing in the animal cells, specifically in liver and kidney, we have to describe that GTP is produced, because ATP and GTP both are energy molecules along with the inorganic phosphate. But when we are talking about other tissues, ordinary tissues, then ATP is released. So succinate, which is 4-carbon compound, is produced, and after the formation of succinate, students, one more energy molecule is released, which is FADH2. And then again what? The dehydrogenation process takes place, removal of hydrogen. इसको हम oxidation process भी कह सकते हैं, because dehydrogenation or oxidation are the same thing. Removal of hydrogen, or oxidation is also removal of hydrogen, but removal of electron is also known as oxidation. So here fumarate compound is produced, and you can see there is a double bond 2 carbon atoms. This is also 4 carbon compound. Obviously, the enzymes are what I have to say, the specific enzymes are working here to convert one compound into another. So, succinate dehydrogenase enzyme is working to convert the succinate into fumarate along with the release of this energy molecule, which is FADH2. Next step is hydration. Here, obviously, the water molecule is added, so double bond is converted into single bond. Here again 4 carbon compound is produced, but the compound name is malate. Right, students, after the formation of malate, you can see here energy molecule is released. So how many energy molecules of NADH are produced? So this is 1, this is 2, and this is 3. It means in the 4th step, NADH2 produce. In the 5th step, one more NADH2 is produced. And in the 9th step, one more NADH2 is produced. So there are 3 NADH molecules are produced in the Krebs cycle. And if you are looking FADH2 molecule, so just one FADH2 molecule is produced, which is produced in the 6th step. And then students, you are looking 1 GTP or ATP, which is produced after the 5th step. So you can see after the 5th step, the after the formation of succinyl coenzyme A, 1 GTP molecule is produced and 2 carbon dioxide molecules, they are lost. So after the regeneration of oxaloacetate 4 carbon compound, again acetyl coenzyme is added and citrate synthase in the presence of this enzyme. In the presence of water also, I have to say 6 carbon compound is produced. Here enzymes names are also mentioned in this Krebs cycle. अगर आपको simplest Krebs cycle याद होगी and the steps of the Krebs cycle, if you know, then you are able to explain this whole Krebs cycle. So in this complete Krebs cycle, you can see I have mentioned the name of enzyme also. I have mentioned the production of energy molecules. I have mentioned the liberation of carbon dioxide. Right, students? Hopefully आपको ये Krebs cycle समझ आ गया होगा. आप इस पूरी Krebs cycle को अपने वर्ड्स में एक्सप्लेन कर सकते हैं. ओके स्टूडेंट्स, थैंक यू. अल्लाह हाफिज़.